Song competition affects monoamine levels in sensory and motor forebrain regions of male Lincoln sparrows by Haley Dunster, Sarah Bryant, and Carol Ann Bailey.
In order to find a mate, male animals will change their behavior to be the most attractive option possible. In male Lincoln sparrows, this is done through song. These sparrows are capable of changing their songs over the course of a week in order to boost their competitiveness against other birds. This experiment was conducted to understand how neurons are able to adapt to create more songs so quickly. We looked at different brain regions that are specifically associated with song production and perception to analyze their neuroplasticity, which is regulated by neuroamines. Based on the environment conditions, these monoamines will change to create the appropriate biological response. High-pressure liquid chromatography, in conjunction with electrochemical detection, was used to correlate levels of monoamines and their metabolites in male Lincoln sparrows that had been exposed to different songs with varying levels of challenge. The two main regions analyzed were in the auditory telencephalon. They were the caudomedial mesopallium and the acropallium. These regions helped show differing levels of norepinephrine, the chemical sensitive to song auditorily. Dopamine is measured because it regulates song output. We continued this examination by looking at serotonin in all four brain regions because of its role in perception and behavior. We discovered through this study that males exposed to more difficult songs had higher levels of norepinephrine and lower levels of serotonin.
Animals are forced to adapt to their environment in different ways to ensure survival of the fittest. In order to carry out these adaptations, monoamines in the brain respond to the changing environmental conditions, forming new neural pathways in the process of neuroplasticity. These monoamines are capable of shifting synaptic transmission, in turn altering how aroused, attentive, and motivated something is. Lincoln songbirds actively change their singing to make themselves more competitive compared to other birds, a form of adaptation. In order to be competitive in the search for a mate, a bird should have a long and complex song. Other birds will respond to this competition over a week by creating even more songs. The levels of norepinephrine in the brain were measured to determine the sensitivity of the neurons in the auditory forebrain, while the levels of dopamine were measured to observe the processes of context-specific singing. Serotonin was measured as well, as is important in the perception, aggression, and vocalization aspect of birds. Once complete, the goal is to determine how changes in these monoamines throughout different brain regions has an effect on changes in social behavior.
Figure 1 shows the effect of prior song challenge on singing effort. The mean number of songs males in the more challenging and less challenging treatment group produced the morning after song playback ceased. The difference in song number reflects differences in males' competitive states because singing was not occurring in response to a playback stimulus. 18 male Lincoln sparrows were used with the chamber mimicking their habitat. After the experiment was conducted with different variations of song difficulty, the sparrow brains were harvested and examined using a cryostat and initial stain procedure.
Figure 2 shows the brain regions that were explored in the study, including the caudomedial mesopallium, area X, caudomedial nidopallium, acropallium, and the monoamine centers for the brain, including the locus coeruleus, the ventral tegmental area, and raphe nuclei.
We split the sparrows into two groups, one with more challenging songs and the other with less challenging songs. These difficulty levels were rated based on how receptive a female bird was to them. Using a library of 96 bird songs from the environment, the birds were exposed to a range of songs with different difficulties. The birds listened to 6 songs over the course of a week. Each song that the bird produced after hearing the playback was recorded numerically. After the week, the brains of the sparrows were collected and used to determine the changes in monoamine concentrations. The caudomedial mesopallium and the acropallium were compared. These brain tissues were extracted using micropunches, and the differing concentrations of norepinephrine, dopamine, serotonin, and their primary metabolites were recorded to compare the two experimental groups of less challenging versus more challenging songs.
It was determined that male Lincoln sparrows that were exposed to more challenging songs over a week had higher levels of norepinephrine in their caudal-medial mesopallium in comparison to a sparrow of the same species exposed to songs that were less challenging. It was also noted that those birds exposed to more difficult songs had lower levels of serotonin in the acropallium. We were unable to find any scientifically reliable variations in monoamines in other regions of the brain, even though they were also subjected to the challenging songs. There wasn't a statistical relationship determined between the male's singing effort and singing in the morning before a sacrifice was found either. It cannot be ignored that self-stimulation resulting from a male's singing could potentially have contributed to the recording differences, because singing behavior has a positive association with playback treatment. The monoamines and the metabolites' varying levels are caused by challenge level of the song, but cannot be correlated with recent singing.
Figure 4 shows the monoamine levels and song playback correlation and compares the levels of monoamines and metabolite with the level of song challenge that the Lincoln sparrows were exposed to. This figure allows us to show the levels of norepinephrine and serotonin in the different brain regions and how they differ between levels of song difficulty.
Male Lincoln sparrows that were exposed to more challenging songs had higher levels of norepinephrine metabolite, suggesting that there are higher levels of norepinephrine breakdown in the caudal-medial mesopallium. Lower levels of serotonin in the principal motor nucleus that controls song pathway were observed when more challenging songs were present as well. It was determined that the monoamines in the brain are involved in sensory and motor processes. These same monoamines are responsible for neuroplasticity by altering the excitability of the neuron, which is causing shifts in the behavioral state. The caudal-medial nidopallium stained for dopamine beta hydroxylase, showing a strong correlation with the release of neurotransmitter. Norepinephrine was deemed the neurotransmitter and is important in focusing attention and improving perceptions in sensory brain regions, especially in auditory processing. Neurons in the caudal-medial nidopallium respond to newer neural activity. This means that any differences noticed between the two groups in terms of norepinephrine release occurred sooner than later. It was also noticed that repeatedly playing difficult songs caused serotonin levels to decrease in the song motor control pathway when compared to the repeated playback of less challenging songs. With further investigation, it was learned that decreased serotonin levels occurred in the birds exposed to more difficult levels of song in general. In contrast, there was a lack of change in the levels of dopamine in area X, which was surprising because this area is associated with situational specific songs. Male Lincoln sparrows varied in the number of songs they produced, but did not differ in the quality of the songs. The variations in song development is correlated to the neural activity in the acropallium. This is consistent with the fact that the changes in the serotonin in the acropallium are a function of social experience. Differing levels of serotonin were correlated with song difficulty, but couldn't be correlated with singing behavior before harvesting the brain. Brains with less serotonin were associated with more competitive birds using more difficult songs. And serotonin was also found to be linked with the ability to monitor vocalization across different classes. Our experiment concluded that changing the difficulty of a bird's song is controlled by monoamines in specific brain regions used for song production. Combined with social cues, these form a short-term adaptation that evolves into the long-term using neuroplasticity. Things that we would change when conducting this study in the future in order to improve it would be determining a timeline of the differences in the brain or changing this experiment in order to explore the relationship between the differences in monoamine levels over social context and timelines with the behavioral state. It's also possible to use different bird species in different songs. A flaw could be a difference in monoemergic activity before the experiment was conducted, which would affect the results in neuroplasticity and behavioral shifts. This experiment could not distinguish the duration of when the differences in neuroplasticity and behavioral shifts occurred, so we would have to work to account for those in the future. We could also further analyze how serotonin might contribute to competitive singing by changing serotonin levels and investigating the behavioral changes. Finally, we could look at how a different species and changing the conditions of the environment would affect the results.